Hi all, I am Arun, currently pursuing a master's in Electronics and Computer Science branch at McGill University. Today, I'll be presenting you my research paper. I am presenting you my research paper titled An Accelerometer-based eyeglass to monitor food analysis in free living and in lab environment. Firstly, I'll be introducing the concept, and secondly, I'll explain you the monitoring system where how we have designed our hardware system, and then I'll take you over to the methodologies in which I'll explain you in which all condition we have tested our system. Once we have collected the data, then we'll discuss the data analysis and the feature extraction. Finally, we'll discuss the results and the conclusion. So initially, as we all know, that it is crucial to track dietary habits, which in turn gives added insight in determining the overall well-being of a person. Unhealthy diet is a significant cause of fatal health illnesses like diabetes, heart diseases, and even cancer. Obesity is also another health issue which is increasing day by day in the world, which, when addressed, can be prevented from aggravating to more terminal problems such as stroke, type 2 diabetes, and other diseases stated above. Monitoring food and intake in free living individual is probably the most difficult problem when doctors want to study the food habits of individual. Currently, doctors use methods such as food frequency questionnaires, multimedia diaries, and self-reporting diaries. However, these methods suffer from low accuracy since people are prone to underreporting and miscalculating the food consumed. These methods are tedious and lack robustness. Therefore, there is a quest for the development of a good dietary monitoring system which can accurately collect and present the data. As a result, we came up with an idea of tracking the mastication muscle, temporalis muscle, which is a fan-shaped muscle located on each side of the head, which contracts and relaxes based on the movement of the jaws during chewing. We use accelerometer sensor to track the movement of the temporalis muscle. Accelerometer sensor is a low noise motion sensor, which does not require complex data processing. And as you can see here, this is our monitoring system, and we have used an eyeglass. The eyeglass we have used because the study by the Vision Council of America states that more than 75% of the adults use some sort of eyeglasses. Eyeglass is the principal component of our monitoring system. The eyeglass is the principal component which has the following components in it. We have used an integrated microcontroller that is Atmega 328P and a Bluetooth chip. Along with these two components, we have an analog 3-axis accelerometer unit that is MXR 9500MZB. This accelerometer sensor is attached on top of the microcontroller and the Bluetooth chip to make it compact and lightweight. The accelerometer sensor records the data, the microcontroller unit and the Bluetooth unit access the data and sends it to the host processor and a lithium battery to power our system. In the methodologies to test our system, we actually took two environment in consideration. That is, first we tested our system in the lab environment, then we tested our system in the free living study. For that, we recruited 4 participants for testing the system, and none of the participants had any dental or vision related issue. For free living study, data was recorded for a day by each participant to enable the training of machine learning algorithm. When we saw the data which we collected from these participants, we observed that there is a data imbalance, as the data for the achieving event was very less compared to the data for the non-achieving event. Hence, this leads to an anomaly detection. And as a result, not only accuracy, but also precision, recall are the best parameters to analyze this type of situation. And hence, totally 35.3 hours of data were collected from the 4 participants in free living condition. For in-lab study, we made the participants seated and we provided with food of varying hardnesses like wheat bread, carrot, cucumber, and banana. Before we provide these four foods, we also told them to not achieve anything for the initial time so that we can record the non-achieving event data. As a result, we were able to record a total of 69 minutes of data from these 4 participants. Once we have collected the data, we normalize the magnitude of acceleration, the magnitude of the resultant using the formula, and calculated the magnitude of the resultant using the formula given here. This magnitude R of t was partitioned into time frames of 1 second each. With these minuscule time frames, we were even able to detect small eating events like consumption of snack, chocolates, chips, etc. For each of these time frames, feature listed in table were extracted. We extracted time domain features like minimum, maximum, range, interquartile range, standard deviation, variations, skewness, and kurtosis. Frequency domain features like PSD, entropy, and principal frequency. So as you can see, this is a 7.5 hours of a data of a particular participant in free living condition. Here, the rectangle A, C, and F represents the achieving event data, and B represents drinking water, D represents playing video game, E represents routine workout. As you can see, E and F looks similar, but our algorithm does a very good job of differentiating them. The data collected in both free living and in-lab conditions were partitioned into 80-20 ratio, with 20% of the collected data set as test set and 80% of the collected data set as training data set. While dealing with free living, the number of data points collected from achieving points are comparatively lower, as I have said earlier. As a result, we calculated precision, recall, and F1 score. For free living condition, support vector machine, KNN, logistic regression, and decision tree classifiers were applied on the test data set to detect achieving event. Among 4 methods, KNN with k is equal to 5 produced the best result in terms of accuracy and other factors. KNN provided an accuracy of 86% with 82.14% of precision, 85.49% of recall, and 82.23% of F1 score. And the other algorithms also provided comparatively better results, as you can see in the table. This is a 20 minutes of test data of a particular participant in the in-lab setting. As you can see, we can actually differentiate between non-achieving and achieving event, but it is hard for us to differentiate between the different types of food achieving event. But our algorithm does a very great job of differentiating them with a good accuracy. The KNN classifier actually distinguishes them, and it gives us a very good accuracy. For achieving detection, SVM and KNN produced an accuracy of 99.37 and 97% respectively. And we got an accuracy of 88%. We got an accuracy of 88% with KNN and 85.37% with SVM to differentiate between bread, carrot, cucumber, and banana. The main idea of conducting the in-lab study was actually to see how we can differentiate between different types of food which has different hardnesses. And we got a very good accuracy from that. To conclude, we have designed an accelerometer-based eyeglass for wireless monitoring of food intake. Achieving detection using accelerometer is actually very simple, and the system provides us a better results compared to other methods. This system is lightweight, reliable, and portable for continuous monitoring. Thank you, one and all.